Hello, everybody. Welcome to Science Abounds, Science at Home, where we will discover home lab magic. In this channel, we will be performing experiments demonstrating scientific principles, concepts, and reactions in chemistry, biology, microbiology, microscopy, and even molecular biology. Welcome to my channel. Hello, everybody, and welcome to another episode of Science Abounds, Science at Home, where we will discover home lab magic. In today's episode, we're going to be looking at the microscopy of tardigrades, and I'm going to dedicate this whole video just on looking at tardigrades, also more affectionately known as water bears. And they are fascinating microorganisms. And I just get a lot of pleasure by just sitting and watching them play and eat while looking through the microscope. Tardigrades are, like I said, very fascinating microorganisms and were named water bears by Thomas Henry Huxley in 1869 because of their inherent poring that somewhat resembles that of the clumsy actions of a circus bear. So why don't we now set up the microscope to look at these fascinating creatures and learn a little bit more about them. Okay, so I have a culture or a sampling of tardigrades that I got, and you can either get them or search for them yourself. Tardigrades are typically found in areas of water around lichen, algae, moss, etc. So they're easy to find in any areas where there's a collection of water and vegetation debris, which they typically feed on. But you can also purchase cultures, live living cultures, from a variety of biological suppliers. And if you look in my video description at the end of the video, you will see I put a list of all my suppliers that I use all the time for all my home laboratory needs, and all the experiments that I perform, I purchase through one or two of these biological suppliers. So in order for me to look at the tardigrades under the microscope, I typically don't use a microscope slide and cover slip. I don't want my microorganism to be squashed or inhibited in any way. So what you can purchase are these what they call demo slides. Okay, and what they are is just basically a vessel to put your culture in, and then it has a very thin capsule at the bottom. And what it does is it allows you to view under the microscope free living microorganisms without impeding their movement or squashing them. And there's an adapter that you put on your microscope stage. This is the adapter. And basically, the demo slide slips in. You slip it in like so. And this viewing area is what you view under the microscope. And the only drawback is you cannot go under high magnification. You can only really use low magnification, but that usually is enough, and it certainly is enough for when you're looking at tardigrades. So this is what I'm going to use today to view the tardigrades. So I'm going to fix the slide onto my microscope stage. Like so. And then I'm going to put a sampling of my culture that I have of tardigrades into my demo slide. So what I'm going to do is I want to pick up, there's some debris in here, some algae, which is used for feeding, which the tardigrades feed on. So I'm just picking up some liquid, and what I do is I put it into my demo slide. I'll put a little bit more liquid, and then I cap it. And what I do is I just want to tap to make sure that the bottom, the viewing area, has liquid in it. Okay, and then I just fit this into the adapter on my microscope, and I can now view my tardigrades or view whatever I have in my microscope slide, in my demo slide. You can use microscope slides if that's what you want. That's fine. But just remember that you could you could accidentally squash the microorganisms, and tardigrades are quite large. So in order to avoid that, I don't like putting them on a slide with a cover slip. I did, for a higher magnification, I did a water mount on a microscope slide, but I didn't place a cover slip on it. But I was able to go far down enough to focus on a higher magnification. And also, please bear in mind that these are whole organisms, 3D. They're translucent, a lot of them. We have a lot of baby tardigrades in my culture, so they are kind of small and translucent. So focusing and getting a whole picture of the tardigrade can be a challenge, but it's part of the fun. So when you see the video, you'll see the images, and you'll see me constantly focusing in and out to get a proper, good picture of the tardigrade. Okay, so I hope you enjoy watching these wonderful creatures as much as I do. So let's take a look. Tardigrades, also known as water bears or moss piglets, are microscopic water-dwelling animals famous for their incredible resilience. These tiny creatures range from about 0.3 to 0.5 millimeters in length and are most often found in moss, lichen, leaf litter, or the sediment at the bottom of bodies of water. So what makes tardigrades so cool? Tardigrades can survive in a state called cryptobiosis, where they essentially shut down their metabolism by losing nearly all of their water content. This allows them to endure extreme conditions, such as extreme temperatures, from nearly absolute zero to over 150 degrees Celsius. They can also withstand doses of radiation that will kill most other life forms. They also survive high pressures, both deep sea and vacuum like space conditions. And they can survive dehydration in desiccated states for decades. Tardigrades made history by being the first known animal to survive in space. In 2007, researchers sent them into space on a European Space Agency mission. After being exposed to the vacuum and radiation of space, many of the tardigrades were able to revive when they returned to Earth. They achieved this survival through a protein called trehalase, which helps preserve the structure of their cells when they dehydrate. They can also form a protective layer around their bodies known as a ton, which makes them even more resistant to harsh conditions. Tardigrades are ancient creatures with fossils dating back over 500 million years. The evolutionary history and unique biological processes make them an interesting subject of study in evolutionary biology and extremophiles. Because of their hardiness and ability to survive extreme conditions, tardigrades are studied in a variety of fields, including space biology, for understanding how life might survive in extreme environments. In cryobiology, they're studied for their ability to withstand freezing and desiccation, and this has led to research into preservation techniques. And in radiobiology, they are studied for their resistance to radiation, and this is a key topic of study. Despite their tiny size, tardigrades have a complex anatomy. They have a head with a mouth that has stylets for piercing plant or animal cells. They are often herbivores or feed on other small organisms. The green coloration of their body is from the ingestion of algae material, and because they are typically translucent, any of the algae or plantation that they ingest can be seen in their body. They have clawed legs, and these are used for locomotion. And they also have a nervous system. They have a digestive tract and reproductive organs, though are highly simplified compared to larger organisms. Their bodies have three main layers. First is the epidermis or outer layer, provides protection and contains specialized cells that produce the protective ton during cryptobiosis. The middle layer, or known as the mesoderm, contains muscles and circulatory elements. Tardigrades don't have a true circulatory system, but they rely on a fluid-filled body cavity for the transport of nutrients and gases. And then they have an inner layer called the endoderm, and this contains their internal organs, including a simple digestive system and a reproductive system. The ability to survive in extreme conditions is the hallmark of tardigrade biology. This phenomenon is called cryptobiosis, where tardigrades enter a state of suspended animation, effectively stopping all metabolic processes. When faced with unfavorable conditions like extreme dehydration or freezing, tardigrades retract their limbs and curl into a ball, forming a ton. During this state, their metabolism drops to near zero, and they lose almost all of their water content. Some can survive with just 3% water in their bodies. In the ton state, tardigrades can withstand temperatures ranging from absolute zero to over 150 degrees Celsius, as well as the vacuum of space, intense radiation, and dehydration. Once favorable conditions return, tardigrades can rehydrate and come back to life, resuming normal metabolic activity. Trehalose, one of the key molecules involved in cryptobiosis, and is a sugar that helps preserve cellular structures during desiccation. There are also protective proteins, and they produce proteins like DSUP, damage suppressor protein, which helps protect the DNA from radiation-induced damage. This is especially important for their ability to survive high radiation environments, as seen in space experiments. Tardigrades are primarily herbivores, but some species are also carnivorous or omnivorous. Tardigrades feed by inserting their needle-like mouthparts, called stylets, into plant cells, algae, or small invertebrates. They then suck out the cell contents or the fluids of other small creatures. The mouth is often covered by a buccal tube, which helps in the feeding process. Tardigrades have both sexual and asexual reproduction, depending on the species. In sexual reproduction, tardigrades have separate sexes, but many species also have hermaphroditic individuals. The males typically fertilize the eggs of the females by external fertilization. In asexual reproduction, some tardigrades reproduce by parthenogenesis, a form of asexual reproduction, where females produce offspring without fertilization. Tardigrade eggs are resistant to extreme conditions and are often involved in their survival. These eggs can endure high radiation and dehydration, which is essential when the adult tardigrade enters cryptobiosis. Tardigrades live in a variety of environments, but are particularly common in moist habitats like mosses, lichen, leaf litter, and soil. Some species are also found in marine environments, often on the surface of seaweed or in aquatic sediments. In terrestrial environments, tardigrades are more likely to enter cryptobiosis due to dehydration. They rely on moisture from rain, dew, or humid conditions to remain active. In aquatic environments, tardigrades remain in a hydrated state, actively feeding and reproducing. When conditions dry up, they can enter cryptobiosis to survive until the water returns. Tardigrades belong to their own phylum, Tardigrada, and are considered one of the oldest groups of animals. Fossils of tardigrades date back to over 500 million years ago, making them a crucial subject for studying evolutionary biology. Tardigrades are thought to be closely related to arthropods like insects, spiders, and crustaceans, based on genetic and developmental similarities, but they have a distinct evolutionary lineage. Wow, what fascinating creatures they are. They're such beauty as well. Please, I hope you enjoyed watching the tardigrades and watching these microorganisms, and I hope you learned a little bit more about them than perhaps you did before. Please come by. I plan to do many more microscopy videos just like this, looking at a whole array of microorganisms and fungi, bacteria, protozoa, protists, slime molds, all these wonderful, beautiful microorganisms, and exploring the world of microscopy because it is truly beautiful. So please watch out for future videos in microscopy if that's a particular area that you like. Also, please do subscribe. Thank you to all my subscribers who have already subscribed. Thank you for your support. It means so much to me. If you're viewing this video, please go and hit the subscribe and like buttons. It's free, doesn't cost you anything, and it shows me your support and helps continue supporting this channel, making many more exciting home experiments. Please also leave me a comment, send me an email. The email you can send me, you can send me questions about science. If you have any questions about science, about my channel, what you would like to see, whether you want to do home experiments yourself and need some advice from me, I'm perfectly happy. Also, send me your photos and videos of you doing your home experiments. I want to truly build up a home lab community. So, with that, until next time, keep on experimenting. Goodbye, everybody.